When you look out over the Stanford campus, most of what your eye sees is a dense patchwork of plants, living, breathing, ever-growing plants. Plants like these provide the food on our plates, the wood on these walls and ceilings, even the very oxygen we breathe. I'm a plant biologist, and to me, one of the most amazing facts about plants is that their bodies are built mostly out of air. The heft of a tree comes not from the soil, not from the rain, but from carbon dioxide that's pulled in from the atmosphere. To absorb all that carbon, plants use these tiny pores that cover every surface of their leaves. And you can see them. If you look at a growing leaf under a microscope, you'll see a picture, like that one on the top left, with two kinds of cells, a sea of immature stem cells in gray, and among them, in purple, those tiny pores through which carbon dioxide enters the plant body and oxygen escapes. It's through those pores, numbering in the billions or trillions in a typical tree, that plants breathe. So, how are these pores made? The object of my dissertation was to understand how that sea of immature stem cells gives rise to these pores. We started this project by simply looking, and we noticed something funny. It turns out that among all of those stem cells, the only ones that ever turn into pores were the smallest ones. That observation led to a hypothesis that the cells may be on a sort of cycle, illustrated here, where on each pass, a cell starts by checking its size. And if it's a big cell, it keeps riding that cycle, divides, gets smaller, tries again. But if it's really, really small, it exits the cycle and turns into a pore. So that was the idea, but in science, ideas are the easy part. What's hard is testing them. To test this model, we turned to a favorite tool in the biologist's toolkit, the genetic mutant. For a biologist, a mutant isn't defective, it's an opportunity to change one aspect of biology and observe the consequences. In our case, we found mutants that change the size of those stem cells. And, just as our model predicted, if you make them really big, they stay on that cycle for much longer. But if you start them out really small, they come in, check their size, and exit on their very first pass. So by changing the size of these cells, we can control their behavior and thereby the production of these pores. Our work highlights how out all around us on this campus, and in forests and farmers' fields, and in our own gardens, tiny little stem cells are making decisions about how to build a robust and functional leaf. And I believe that in a future where food security and climate resilience depend on smarter and more adaptable crops, we can use these discoveries to not just understand the behavior of stem cells, but to control how plants breathe and grow. Thank you.